kind of. Oh man, this sucks. Can you hear me now? In the back? Yeah? Over there? It got better. I'll just hold it like this. Yeah. Okay, cool. So I hope you were all able to find the practice test online. Like I said on Monday, I'm more than happy to go over it and talk about a few things while I go through the whole test. Or if you want me to, I can just answer questions and just go from there like we did on Monday, which went pretty good because we talked about a few important things in my opinion. So I don't know if, are there any questions or things you would like me to go over? Or do you just want me to go through the test? Okay, cool. Awesome. So number 1 says, which of the following is not characteristic for inflammation? An activation of macrophages, increased vascular permeability, vasodilation, pain, and influx of leukocytes. Which one do you think is not characteristic of inflammation? A? Yeah. Why is it not a good thing that you inactivate macrophages when you have inflammation? What do macrophages do? Huh? Yeah, they're the ones who will eat everything up. They'll secrete cytokines, remember? Like TNF alpha, IL-6, CXCL8, and so on. Those will all cause inflammation that will eventually call also other cells like neutrophils, NK cells, and so on. And that's how you get the immune response. So for number 1 is going to be A. I'll go ahead and write the answers here. And then I'll still upload them as soon as we're done here. I'll go ahead and upload the answers, okay? But I'll write them down here just so that we can keep track of everything. So which of the following lymphocytes has an orange-red stained granules and is specialized in fighting parasitic infections? You should have seen this question at least a couple of times in his tests. And hopefully you got it right all 5 times, right? So anyone can tell me which one of the following lymphocytes has? None, right? Yeah. Because remember, and I'll write it over here. Hold on. Let's do this so you can see the whole. Okay. There you go. Remember the hematopoietic tree, the one with all the different cells that you studied at the beginning of the class? You had the stem cell. You had the lymphoid precursor that gave rise to T cells, B cells, and NK cells. And then you had the myeloid precursor that gave rise to Ben, monocytes, which turn into what? Macrophages, yeah. When they differentiate and they turn into macrophages when they get into the tissue. So will you be able to see macrophages going back in circulation ever for this class, for the purposes of this test? No. Okay, no. Macrophages stay strictly in your tissues. One other cell that does go back to circulation are dendritic cells. Remember, dendritic cells went back to the lymph node, and then from Langerhans cells, they turn into mature dendritic cells with B7, MHC class II, and activated T cells. Remember? Yes, kind of? Okay. So then monocytes, macrophages, and we also had dendritic cells. Cool. Awesome. Can you see well? Kind of. There you go. All right. So then for number 2, it's going to be E. So pyogenic refers to the body's production of heat to fight infections. What does pyo mean? Pus, right? And pyro means heat. So just remember those things, because those are little details that you might not see when you're taking the 100 questions test because you're in a hurry. So just make sure you read word by word, mark the keywords that you have on a question, just so that you actually answer it correctly. Because that's the way you just not make it. I mean, you lose points this way if you don't pay attention to what's there. So it's pyogenic refers to the pus production, not the heat production. So then question 3 is false. So it's B. All right. 4 is going into the whole complement cascade. So it's talking about which of the protein will activate the classical pathway, will initiate the classical pathway. Is it C1q, C3b, C1s, both C1q and C1s, or none of the above? 4, right? I mean C, which is C1s. Who here wants me to go over complement? Okay, cool. I'll go over complement. So does that bother you? No? No, I have. I brought this time. I'll just use another one. So remember, see up here, yes. We have alternative pathway. Starts with which protein? You guys can talk. It's okay, I promise. You won't. Which protein starts the alternative pathway? What does it start with? Anyone? Yeah, right. C3. Awesome. Thank you. See, you should all be more like her. She's answering stuff. Thank you. So we have C3 gets hydrolyzed into IC3, and then factor D comes in. Factor B, sorry, comes in, and factor D will cleave B into BA and BB. So we're going to end up having IC3 big B, little b, plus BA, which goes away and we don't care about what BA is for. And then we end up having this, which is the soluble C3 convertase of the alternative pathway, right? Is this attached already to the surface of the pathogen, or is it going to be floating around somewhere near the pathogen? If it's soluble, hint, hint. It's floating around, right? When this IC3, we're going to have again C3, right? And you're going to have IC3 big B little b cut IC3 into, cut C3 into C3b and C3a. Then this is going to go to the pathogen, right? and then it can join again with factor B, then factor D comes in and cuts the B into BA and BB, and you're going to have, which is the C3 convertase of the alternative pathway. So once the soluble C3 convertase is formed, you're going to have the soluble convertase cut C3 again that's still floating around near a pathogen and a tissue or whatever, and it's going to cut it into C3A and C3B, and then C3B can, join another B, and B will be cut by factor D into C3 little b, big B, little b, which is going to be your C3 convertase of the alternative pathway. Remember, C3A is very important. Does anybody remember why? What is that? Yes, it causes, if somebody ever offers you an injection of C3A and C5A IV, just say no, because you're going to die unless you have an EpiPen, okay? So don't do that. But yeah, it's an anaphylatoxin. Anaphylatoxin. Cool, awesome. Oh, by the way, sorry about the handwriting part of everything. I apologize, but it's the best I can do. So then we also have the classical pathway. Which is pretty cool because it starts after the alternative, but it ends up making the alternative pathway stronger in a way, right? Because it ends up producing C3, which can turn into more C3 convertase and so on. So the classical pathway, we will need 1 IgM, 2 IgGs, or 1 CRP, in which your C1q protein will bind to, and then it's going to start cutting C4 and C2, right? Into C4A and C4B, sorry, and C2A, right? And then when these two join together, you're going to have C4B, 2A, which is the, oh. Okay, cool. Sorry about that. This is the classical C3 convertase, right? Makes sense? And then this one can cut more C3, and C3 can in turn bind to factor B and so on and blah, 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 right? That's why it's important. Which one happens first? Is it the alternative or classical pathway? Alternative, right? And then the classical happens. Cool. And then one more thing. We also have the MAC, which is the membrane attack complex, which can be formed by both alternative and classical pathways. With the alternative, we're going to have C3b, big B, little b, binding to another C3b, right? So if this cuts another C3 into C3b into C3b, instead of landing nearby, it actually attaches to the whole convertase, it's going to turn into the C5 convertase of the alternative pathway, which is C3b2, big B, little b, which in turn will cut C5 and, recruit C6 to C9, I think, and just do the whole membrane attack complex, which is the big hole in the bacteria, right? That will kill your bacteria. And then the same thing happens, well, similar thing happens with classical, where you have the C4B2A plus another C3B, which turns into C4B2A 3B, right? And this will cut C5, which will recruit C6, C7, C8, C9, and so on. Make sense? Any questions? There you go. Cool. Oh, I'm keeping all these, and I'll put them over here, so at the end, if you don't catch something I write, you can just take pictures of it, okay? All right. Cool. So we said question 4 was C1s, which is option C. Oh, and so with C1, the protein C1 is divided into 3 subunit. So C1 has C1q, C1r, and C1s. C1q, when it binds to the surface, well, when it binds to the IgM, IgG, or CRP, will activate C1r, and C1r will activate C1s. And C1s is the one that, I guess I should change this here. Right? C1s is going to be the one who will actually be doing the whole enzymatic activity of cutting stuff. All right? Cool. So which one is the receptor for C3b? CR1, right? What about C3d? D as in David. CR2, right? Where did we see the CR2 receptor? We saw it recently, remember? With B cells? Yeah? It was binding, the CR2 was binding to the whole complex to activate B1 cells along with the IG, with the BCR, right? Cool. So then 5 is going to be A. If you have any questions along the way, just, please let me know. I'll stop and answer them. No problem. So number 6 is asking about the Tolllike receptor. Remember the Tolllike receptors? You studied at the first protein cascade, signaling cascade you studied. It's something very, very specific for a final, like a cumulative final, but it doesn't hurt if you know something like this. That's why I put it on the test to remind you. But 6 is asking what extracellular proteins does a Tolllike receptor need to be able to start the cytokine producing cascade? Is it MD2, CD14, or LBP, or all of the above? All of them, right? So it's a whole complex along with the Tolllike receptor, which will start the cascade with IRAK4, MYD88, and so on, which leads to NF-kappa B. It's a transcription factor you saw multiple times, which if I was the professor, since you saw it about 4 times, I think, or 5, I would definitely ask something about it. We'll start the sequence, the production of cytokines that will lead to inflammation, remember? And really quick, this is question 6. We're going to have the TLR. Let's say it's a bacteria, and TLR4 recognizes LPS, right? So TLR4 will activate the signaling cascade, which will turn on NF-kappa B and will release cytokines, which are TNF alpha, IL-1, IL-6, CXCL8, and IL-12, right? These first three, those are the those are the pyrogenic cytokines, just for you to remember. Those are the ones that will cause vasodilation. They are going to increase permeability of the capillaries and so on. CXCL8 will call in your neutrophils, remember? And thanks to the vasodilation that these cause, they'll be able to enter the site of infection and so on. So yeah. Good review. Good times, right? Cool. So for 6 is going to be all of the above. 7 is asking about identifying the mismatched pair. So it has ICAM1 with LFA1, CD14 with LPS, and K cells with INF gamma, and selectin with tight binding. Which one do you think won't belong together? D, right? Yeah, thank you. So it's the selectin is involved not in tight binding. That would be ICAM1 and LFA1, for example. The selectin is actually involved with the rolling adhesion when the neutrophil is literally rolling throughout the capillary, right? And then CD14 is part of the Tolllike receptor, and then NK cells are the ones who secrete INF gamma. All right, so 7 is going to be D. So then catalase is the enzyme used by neutrophil to produce hydrogen peroxide. True or false? False, right? It's the one that breaks down hydrogen peroxide into water and oxygen, okay? Where can you find catalase? In which cell? Remember? Neutrophils, do you remember? They had granules in them. The primary, secondary, and the gelatinase one? No, so let me write this down as well. So this is just a reminder of random stuff that's throughout the whole class, that it's just stuff that it's good at if you know it. This is what? Question 8. So neutrophils have primary granules, which are called xerophilic, and secondary, which are called specific. And then you have also tertiary, which are called, or they have gelatinase. Cool. So, all right. All right, so then number 9, INF alpha and beta are produced by which cells? Only NK, only T cells, or all nucleated cells, A and B, none of the above? All nucleated, right? Remember? Those were produced when a cell was invaded by a virus. They would secrete INF alpha and beta, and then NK cells would be the ones who secrete the INF gamma. Okay? So then 8 is false, 9 is C. All right. So one binding site of any Ig is composed by 1 hypervariable region, 2, 3, or 6 hypervariable regions. It's 6, right? do we all remember why? Those of you who came on Monday should at least remember. Does anybody that doesn't remember why? Okay, I'll go over it then. No worries. So this is for question 10. So remember, your antibody looks like this, right? This could be any BCR or any secreted antibody that is secreted as a monomer, right? You have the, oh, I can use colors, that's right. I forgot. You have your heavy chain, and you also have your light chain, right? Sorry, I was never the artistic kid, so that's why my drawings are not so good, but at least you understand. Anyways, the binding set is right here, right? 1 and 2. So, for example, one IgG, secreted IgG will have 2 binding sites, while a secreted IgA will have 4 binding sites because it's a dimer, and a secreted IgM will have 12, right? Sorry, 10, because it's 5. If you zoom in into this, you're going to have this big structure. You can see it good. So this is heavy chain, and this is the light chain. And remember that you'll have these, at least this is how they're drawn in your book, and they're called HV1, HV2, HV3, and the same thing goes over here. 2, HV1, right? Each one of these get their diversity from your V, D, and J genes. So for example, for the heavy chain, so this is heavy, this is light. So for your heavy chain, the hypervariable regions 1 and hypervariable region 2, they'll get their diversity by the V segments. While the light chain, and then the HV3 gets the diversity from the VD, D, and DJ segments, right? And then the light chain, the same thing goes for HV1 and 2. It's going to be the V segments. And then the HV3 is going to be the VJ. Oh, I keep, sorry.
Do you know how to fix it? Go down.
There you go. My bad, sorry. And then the HV3 is going to be the VJ junction, right? This just goes over a little bit more from the question, but it's good review. And so yeah, so one binding set is composed by 1, 2, 3, 4, 5, 6 hypervariable regions, because it's 3 hypervariable regions of the heavy chain and 3 hypervariable regions of the light chain. So then so if you ask for how many hypervariable regions an IgM will have, for example, which I think he asked that question in previous tests, you're going to have 60, right? Does it make sense? Yeah? Okay, cool. Because remember, the IgM has 10 binding sites, so 10 times 6 is 60. I'm sorry? Huh? Oh, okay, cool. Cool. Any other questions about this? I think this is a concept that's very important to understand. So just make sure you have this down. This was question 10, is going to be D. All right. Oh, there you go. I just went over this. So the third hypervariable region of the light chain, CDR3, is determined by the VJ junction. We just went over this right here. So then 11 is going to be C. Question 12 asks whether an antibody is secreted or membrane bound. That's determining during the RNA splicing. Do we all agree? Does somebody would like me to go over why? No? Cool. Okay, cool. Awesome. So let me just write it down. So 12 C. All right. So remember we have DNA that codes for proteins. DNA is turned into RNA, and RNAs are then read and turned into proteins, right? The DNA for antibodies or even the TCRs, it's all the same thing. It's pretty much the same basic concept. You have your genes that code for your VDJ, your VJ, your isotypes, so Ig gamma, lambda, and so on, depending on what you're talking about, if it's heavy chain or light chain. But then in your RNA, when you're producing your antibody, you're going to be able to do you're going to be able to choose between IgM and IgD, sorry, and choose if it's secreted or membrane bound. And this is just because of the way your genes are coded. So remember, let's say this is the heavy chain that we're doing, because the heavy chain, if we look at the structure of the heavy chain right here, that's where the surface would be, and that's where the whole antibody attaches to your cell. If you look at the DNA, you're going to have your VDJ genes, and then you're going to have your isotypes. So which one's the first isotype we always secrete? IgM, right? We're going to have IgM. And then the second one always is going to be IgD. So this would be mu and delta. All right, my Greek letters kind of suck, so that's why I just use regular letters. And then you have, let's say, IgG, for example, and so on. So what happens is that this is DNA. Whenever you want to, let's say it's your B cell, once it finished to go through the whole somatic recombination, but it's still an immature B cell. It still needs to produce its IgM and IgD and put it on the surface, right? So it's going to need to transcribe everything to RNA, from the DNA to RNA. It's going to need the VDJ segments, because that's going to be where the binding site is. It's going to need both IgM and IgD, because remember, naive immature B cells, they're going to have both IgM and IgD on the surface. So what happens is that when we go from DNA to RNA, we're going to have still your V, D, and J segments. And then we're going to transcribe your IgM gene and your IgD gene. And then we're also going to add a little part where you can choose whether it's secreted or membrane bound, right? So then all you have to do once you're going through the whole, once you have to create the protein, I mean not you, the B cell, all you have to do is just decide which part to cut out from your RNA. So that's what RNA splicing is. So if you want a membrane bound IgM, for example, you're going to cut this part out, the secreted part, and you're going to cut this IgD part. And you just, read that through your ribosomes, and boom, you have your heavy chain that's membrane bound. And then you add the light chain, and you're good to go. Does it make sense? Yes? Cool. So this was question 12, right? Yeah, and that's RNA splicing. So then 13 is the whole somatic hypermutation part, in which we do isotype switching and affinity maturation. And the somatic hypermutation, you're going to have AID involved, right? Does it make sense? Yes? And AID is going to be the one who also switches your isotypes, because you're going to have what we call switch regions in between each isotype, where AID can just land and just cut whatever is in between the isotype you want and the VDJ. So then 13 is going to be C as well. All right, there you go. T cells with the TCR. So once a T cell has rearranged the TCR, which of the following membrane proteins will the TCR need to leave the endoplasmic reticulum to get to its final destination, which is the surface of the T cell? Is it going to be 1 IgA, 1 IgB, 2 epsilon, 1 gamma, and 2 deltas? No. Ig alpha and Ig beta, those are going to be the co-receptors for B cells, remember, okay? It's going to be, it's going to be the D, so it's going to be 2 epsilon, 1 gamma, 1 delta, and 2 zeta chains, right? And remember, that's the next question, but remember that the zeta chains are very important in TCRs, just because they're the ones who will help to send your signal down to the nucleus of the T cell whenever a T cell actually gets to read an MHC plus peptide, so that it can get activated, okay? So 14 is D. 15, I'll read it, but I just answered the question. It says that the importance of the zeta chain is in A, their key role in stabilizing the TCR. B, these proteins have a role in transducing a signal from the extracellular space to the intracellular space. C, their role in aiding TCR when interacting with the MHC. D, without zeta chains, ZAP70 would not phosphorylate, and activation of T cells would be impaired, and then B and D. And there's two right ones, which is B and D. So they're the ones who send the signal from the extracellular space to the intracellular space, because they're the one that activate ZAP70, and D is also correct. All right. Can anyone tell me what transcription factor or factors are activated when a T cell is activated? Huh? Yeah, so can anyone tell me which transcription factors are activated when your T cell is activated? So when a T cell can actually read something off of a dendritic cell, MHC plus peptide, it needs transcription factors. That's a whole chart you had to study, where you had ZAP70 activating PLC gamma cutting, something into IP3. I don't think I'm not the professor, so I don't know the test. I haven't seen the test. But things this specific, it's good to know them because it's extra points, but if you don't know the general stuff, make sure you know the general stuff first before you go so specific. Anyways, back to this. Anyone can tell me the transcription factors? Yeah, I call it MFAT, NF, so just side note over here, it was NFAT, NF, kappa B, and AP1. The one we saw several times is NF kappa B. Okay, so just remember, if you can, if you are doing well in the class and you can remember these kind of details, know them. And I just told you this because, like I said, NF kappa B is something that keeps coming back in this class. So I just want you to be ready if there's something like that. Oh, that's AP1. Got it? Yeah? Cool. Oh, was there any questions there? No? Or was it just my bad handwriting? Okay, cool. So then 15 is going to be E. And 16. So we go into MHC1. So MHC1 has 2 transmembrane chains with 2 alpha chains and 2 beta chains. That's wrong, right? Because MHC1 is the one that has 1 transmembrane chain and 3 subunits, right? So B is correct because it's 1 transmembrane chain, 3 alpha chains, and 1 beta 2 microglobulin, right? So hold on one second. And then remember that the beta 2 so I'll read them all. C says has 1 alpha 2, alpha 1, 1 alpha 2 that will form the peptide binding site. And then D says has an alpha 2, a beta 2 microglobulin that also has a transmembrane chain. So B and C are correct because alpha 1 and 2 are the ones on the top that will create the peptide binding site. And then D is not correct because the beta 2 microglobulin is just there to support the whole thing. It's not going to bind to your cell. So if we had to draw it really quick, it would be, this is 16. You would have surface of the cell, alpha 2 and alpha 1. This, ignore this line, it's supposed to be one single thing. And then that's the beta 2 microglobulin, remember? And then the MHC1, MHC2, sorry, it's easier to remember. Right? And this is going to be alpha 1, alpha 2, beta 1, beta 2. So oh man, I'm sorry. There you go. Cool. All right, so 16 is going to be E. So the maximum number of HLA genes that Professor Weigel could receive from his parents in the event that he's homozygous for MHC1 locus and heterozygous for his MHC2 genes is 3, 6, 9, 12, or pi. 9, right? Do we all understand why? Was there anyone that wants me to go over it? Yes? Cool. So this is 17. if you have a question like this and you get confused, you like start blanking, you can just do this easy chart and remember it. So you have MHC1 and MHC2, and then you can have you can be either heterozygous or homozygous, right? So if you're heterozygous for MHC1, you can have up to 6 genes. If you're homozygous, you can have up to 3 genes. And the same thing is for MHC2. Right? So this example says that he is homozygous for MHC1, so you have 3 genes, and then heterozygous for MHC2. So 3 plus 6, 9. Now, if we were to say heterozygous for both, you have 12. If you're homozygous for both, you have 6. It's really easy. All right? All right, so this is 17C, right? Yeah. Cool. Then question 18 says that a mutation in the MHC locus will affect the beta-2 microglobulin gene. Is that true or false? False, right? That's the beta-2 microglobulin is not encoded in your HLA genes. That's just another specific thing I just put there for you guys to remember. I think it was even a question on the test. I don't remember, but just for you guys to know, just in case. Cool. So then 19 is saying that when interacting with a TCR, the peptide littered on the MHC will mainly interact with the blank region of the TCR, while the blank and blank regions will interact and recognize the MHC. Does anybody remember which CDRs recognize what? I see some shaking. So it's going to be CDR1 and 2. I'm sorry. Let's do it in order. CDR3 will interact with the peptide, and CDR1 and 2 will interact with the MHC, right? So when you are going through your positive and negative selection processes in the thymus, where your T cells are going through the whole maturation process, positive selection is really testing whether your CDRs 1 and 2 are recognizing the MHC, and negative selection is really testing whether the CDR3 is recognizing self peptides in that case. So if we can draw it really quick. So that's just the top part of your TCR, right? And 1, 2, 3. 1, 2, 3. So there you go. And then you're also going to have an MHC TCR, right? Just imagine that like this is where the T cell would be, and over here you would have something like a dendritic cell carrying an antigen, for example, peptide, sorry. And this is where your peptide would go. So your HV1 and 2 will recognize the MHC, while your third hypervariable regions will recognize the peptide. It's that easy. Does it make sense with the drawing? If you go back on your PowerPoints, there's actually some pretty cool pictures on, it's like a view from the top, and you can see how the peptides, how the MHC and TCR kind of interact, and they're kind of crooked too. But this is a simplified version of what's on the PowerPoints. All right. So then the correct one is going to be, for 19, is going to be B. I'll just write it over here. All right, so then 20 says, it's asking about SID, which is severe combined immunodeficiency. And it's saying which statement is true about SID. So it says T cells are functional because with SID, TCR recombination is not affected. B saying that T cells are not functional because with SID, TCR recombination is affected. And then one of the RAG genes is affected, which does not influence the TCR recombination. B and C, or none of the above. So the right one, although some of you would like to put D, it's actually B, because one of the RAG gene is affected, but it does influence TCR recombination. So it's B, basically. So with SID, your RAG genes won't work, so your T cells won't be able to make any working TCRs. All right. So little Jimmy was taken to the doctor because of an upper respiratory infection that he does not seem to be able to clear. He has chronic respiratory infection. His differential blood cell count showed that Jimmy had a normal lymphocyte count, but mapping of his HLA genes showed that there were severe mutations affecting his TAP proteins. Most likely, Jimmy will have no opportunity to load MHC1 molecules with peptides, severe combined immunodeficiency, bare lymphocyte syndrome, A and B, or A and C. So which one do you think Jimmy has? Anyone? I'm sorry? E, right? Yeah, so he has bare lymphocyte syndrome, and you won't be able to load your MHC1 molecules because remember that your TAP proteins are those two proteins that form sort of like a canal between the cytosol and your endoplasmic reticulum, and will carry the peptides from the cytosol to the MHC1. If you don't remember what I'm talking about, you can just go back and I forgot which chapter it was, but the chapter with the MHCs, I think it's like 5 or 6, and the drawing, it's pretty self-explanatory, okay? So 21 is going to be E. Then 22, it's saying HLA-A will present antigens to a CD4 T cell. B will present antigens to a CD8 T cell. C is one of the MHC1 isotype. Other isotypes include HLA-B, HLA-C, and so on. And then A and C and B and C. Anyone remember? No. So it's going to present to CD8 cells because HLA-A is the same as an MHC1 molecule, right? And it's an isotype of MHC1. You're also going to have HLA-B, HLA-C, and so on, right? So then 22 is going to be E. Oh, so then you just have to arrange the correct sequence for the stages of B cells. So I'm not going to read all that. The correct one is going to be D. So it's going to be early pro, late pro, large pre, small pre, immature, and mature. And remember that with your early pro and late pro, large pre and small pre, that's where your whole somatic recombination is happening. When you're at the large pre stage, you're actually testing your heavy chain with the surrogate light chain, which had V pre gamma and lambda 5. Yeah, I think. Can anyone double check me? V pre B, thank you. V pre B and lambda 5. And then your your small pre is going to be the light chain recombination. When you're in a mature B cell, you're going to have only IgM on the surface, and when you're a mature cell, you're going to have both IgM and IgD on the surface. Okay? So 23, we said D. So, oh, I just answered this as well. So a large pre-B cell is distinguished by, it's going to be, hold on, 24E. So it's going to be both B and C. So it's going to be the pre-B cell receptor, which consists of a heavy chain and the surrogate light chain with its Ig alpha and beta on the side. And then the surrogate light chain will test whether the heavy chain is able to bind to the light chain. Do you guys remember that? So on the, this is 24. So you'll have your heavy chain, but instead of your heavy chain, instead of having the light chain already, we'll just have your surrogate light chain. So this is V pre-B lambda 5. And then you're also going to have your Ig alpha and beta. That's it. And then once this happens, you're going to have those positive signals that will stimulate your B cell to start your light chain rearrangement. You're going to have a allelic exclusion because you're going to stop arrangement of the heavy chain, so you don't mess with the other chromosome and create a new another heavy chain, basically. Oh, let me actually add here. And then once you have your light chain done, they're just going to attach to where the surrogate light chain was. All right. All right. So 24. What's that? These? Ig alpha and beta. So hold on. Yeah, please tell me if you don't understand my handwriting, because I know it's bad, so I won't get offended, I promise. I'll just rewrite it. There you go. That's prettier. So then we said 24 was E. 25, we're asking about mature B cell. We'll choose whether to produce IgM or IgD through RNA splicing. Remember, we went over this. Where is it? There it is. RNA splicing. So you're going to have your VDJ genes. It's going to choose between IgM or IgD. And then it's going to be able to choose between secreted or membrane bound. In this case, it's asking about IgM or IgD. So what your cell will do is we'll cut out IgM if it wants to do IgD, or we'll cut out IgD if it wants to do IgM, basically. Make sense? Yeah? Cool. So 25D. I'm sorry? For 25? It's D, right? Oh. Yeah, no problem. Sorry about that. So 26, that's going to be the whole checkpoint thing. So your pre-BCR is also known as your first checkpoint, which is a positive signal that prevents apoptosis, right? Because your surrogate light chain, sorry, I was blanking. Your surrogate light chain is actually a good thing if it can bind to your heavy chain. So if it does, it wants your B cell to continue with the whole process. So it's actually going to, prevent the cell from dying, right? So 26 is going to be C. Any questions so far? No? Cool. So a translocation of your MYC gene will give you superpowers. Not really, unfortunately. Will give you SID, bare lymphocyte syndrome, or Burkitt's lymphoma. Burkitt's lymphoma, right? Yeah. It's a lie that mutations give you superpowers, unfortunately. It would be awesome, though, but it's a lie. So 27B. Cool. So then, this is talking about B1 cells. So remember, B1 cells are those weird B cells that they don't really need activation from T cells. And it says prenatal B1 cells have little diversity because AID is not expressed in the prenatal period. TDT is not expressed. The VDJ junctions are more diverse, or none of the above. So in a question like this, C doesn't make any sense because they have little diversity, so they're not going to have more diverse VD and J junctions, right? And then you have to choose between AID and TDT. So which one's involved in somatic recombination, in making your your BCRs? Is it AID or TDT? TDT, right? Does it make sense? Remember, AID is only involved in somatic hypermutation, not somatic recombination. So the answer is going to be B, right? Because also remember, B1 cells, they don't, yeah. Can you speak up? Oh yeah, so he's asking to go over the differences between somatic hypermutation and somatic recombination. So this is what, 28? So we can just write it here. 28, right? So somatic recombination it will happen in your bone marrow for B cells, and in your thymus for T cells, right? And in somatic recombination, you'll have generation of your BCRs or TCRs, depending whether you're a B or a T cell. So remember that somatic recombination is always first, right? So this is number 1. And then somatic hypermutation, it's actually going to be, it's only going to happen in your B cells, remember? Just because T cells don't really need to do that. And that's where you're just going to have, so somatic hypermutation, that's going to improve your BCR basically. So you're going to have affinity maturation and isotype switching. Make sense? Yeah? Cool. Awesome. So that's basically the difference. And somatic hypermutation will happen in your secondary lymphoid tissue. All right. So if you have a question where Professor Wiggle is asking, somatic hypermutation is the process where BCRs are created, you're going to say no. Yeah? Yeah, so the B1 cells, those are those weird B cells that we said that, first of all, before I go over that, is there any questions about this? No? Okay. Your B1 cells, those are just another type of B cell. So we've been studying B2 cells, which we just call B cells. But we also have another type of B cells called B1s, which are just different. They don't need T cell activation, and they're more common in your fetus, basically. When we're babies, our population of B cells is going to have more B1s than B2s, okay? And what happens is that they're going to have little diversity because they don't have any TDT, or they have low TDT. So they can't really create a lot of junctional diversity, okay? Did it answer the question back there? Yeah? Okay, cool. Awesome. So 28B. So, oh, just so you know, we have an hour left and we're kind of in the middle. I hope we can finish this, but if you guys ask me questions, I'd rather answer your questions. And then if anything, I can just post the answers online for this. So if we don't get to finish the whole thing, don't worry about it, because literally like 5 minutes after we're done with this, I'm going to go sit down really quick and upload everything, okay? And you'll be able to find it on the same page, the ucfstudyunion.wordpress.com, where you found the test that you're using now, okay? Cool. So don't worry about asking questions that might take too long. I don't care. I rather answer your questions and make sure you're ready for the final. So then 29, right? So plasma cells can differentiate directly from activated B cell, isotype switched B cells, hypermutated centrocytes, memory B cells, or all of the above. All of them, right? Because activated B cells can just be turned into plasma cells. Even if it's their first time getting activated, they could just go ahead and secrete IgM, right? Isotype switched B cells, they can also turn into plasma cells or memory cells, one or the two, depending on which cytokines you have. And then hypermutated centrocytes, which is just the fancy word for B cells that were in the germinal center, those can also turn into plasma cells. And memory cells, if they're activated again, they can turn into plasma cells as well, right? Do we all know what a plasma cell is? Yes? Just remember, it's the activated B cell. It's angry, it's big, it secretes antibodies. I think of it as like, who saw the movie Rambo? Well, it's okay. I mean, I know a lot of people, not a lot of you saw it. Just think of Rambo holding like the machine gun shooting out, but instead of having bullets coming out, it's IgMs or IgGs and so on. So it's like a very angry B cell that will be shooting out antibodies, basically. That way you won't never forget what a plasma cell is. Anyways, so 29 is E, right? And if you don't know what I'm talking about with Rambo, if you Google it, I'm sure that's one of the first pictures that will show up. So number 30 is asking, which of the following is not a signal that originates from a successful arrangement of a beta chain and the pre-TCR, the pre-T cell receptor? Stimulating proliferation, stimulates expression of either CD4 or CD8 coreceptors, stops any additional TCR beta chain locus rearrangement, and signals to become permissive for alpha chain rearrangement. So when you're answering something like this, just take a deep breath and think of where this is happening. So for example, for this question, your pre-TCR is going to be happening in the thymus, right? That's where your T cell, your thymocyte, is maturating, and it's trying to create a beta cell and a beta chain and an alpha chain, right? So the pre-TCR is going to be the same as the pre-BCR, right? And that's just where you're testing your beta chain. That's it. So once you test your beta chain, think of what needs to come next, the alpha chain, right? So it's going to stimulate proliferation, because once you do something right with a cell, you want to be able to, make more of yourself. It's going to stop any additional TCR beta chain rearrangement, because once you have the beta chain that's working properly, you don't want to create a new one, because you want that one to be the only type of BCR you're doing. You want to become permissive for alpha chain rearrangement, so that you can start with the alpha chain and complete your TCR. And you will stimulate the expression of both CD4 and CD8, not just CD4 or CD8, because that's the point where your T cell or your thymocyte becomes a double positive, remember, okay? Does it make sense? Oh, never mind. Yeah, so the wrong answer is going to, so B is the right answer. It's the one that does not originate from this whole scenario. Does it make sense? Yes? What is that after? So yeah, so once you have your alpha chain, your T cell goes through positive selection, where we recognize the MHCs. We make sure that the T cells we made so far can recognize MHCs. And once they recognize, for example, an MHC2, your T cell is going to keep all of the CD4s and delete all of the CD8s. If it recognizes an MHC class 1, it's the other way. It keeps all of the CD8s and deletes all of the CD4s. Cool. Answer the question? Any questions so far? No? So 30 we said is B. Once the first chain of the beta chain of the TCR is rearranged and the cell is finished proliferating, then the RAG genes in turn are turned back on. Rearrangement of the alpha chain is induced, and there's a bigger chance that the rearranging thymocyte will become a gamma delta T cell than an alpha beta T cell, or it's A and B or all of the above. Which one do you think is the correct answer? A and B, right? Because only 5% of our gamma delta T cells, of our T cells are gamma delta T cells, right? And the most abundant are going to be the alpha beta T cells. Oh, and also remember that whenever he's talking about T cells in general, he's referring to alpha beta. And whenever he wants to ask you a question about gamma delta, then he's going to say a gamma delta T cell. All right? So this is going to be E. The pre-TCR is composed of a fully formed TCR with both alpha and beta chain, a beta chain and a pre-T alpha, CD3 and zeta chains, A and C, B and C. This is really easy. So it's going to be the beta chain, the pre-T alpha, and then the side chains, the CD3 and the zeta chains, right? Do we all agree? What was your question? When? Whenever, so he's asking when the RAG genes are turned off. That's whenever you have a stopping point. So whenever you check something, so whenever you check the beta chain, you stop your RAG genes. Whenever you finished your alpha chain and you confirm that the alpha chain is working, then that's when you stop them again. So whenever you're done with doing your somatic recombination, you're creating your alpha beta chain or heavy chain or light chain, that's when you stop the RAGs. Yeah, because if you don't, your T cell or B cell will keep on doing somatic recombination, and then you're going to end up having a bunch of different TCR on the surface or different BCRs, which you don't want that. Yeah, good question though. Thank you. Does it make sense for everyone? Yeah, cool. So that's what? E. Man, I don't know how professors do this, like talking for like an hour every day or more hours. My throat is like killing me. It's tough. It really is. Anyways, Rearrangement of the alpha chain for number 33, right? Rearrangement of the alpha chain locus continues throughout the process of positive selection and stops whenever the T cell is positively selected. True, right? Yeah, thank you. So remember that in positive selection, we are testing to see if our TCR can recognize our MHC, because that's the most important thing that a TCR can do. Is first you want it to recognize an MHC that comes from your body, and then second, you want that T cell not to recognize your own peptides, because if not, you get autoimmune disease, and nobody wants that. So in order to see if you can recognize MHCs, you go through positive selection. And then in order to see if you do not recognize self-peptides, you go through negative selection. And when you're going through positive selection, you're actually going to keep your RAG genes on, like you were asking, because if your T cell is not able to recognize an MHC right away, it can just try to see if it can create a new alpha chain and see if that new one can actually recognize the MHC. And then if nothing happens after about 4 days, then your T cell dies, basically. Just fun fact, in your positive selection process, I think we kill like 98% of the thymocytes that we create. So it's a pretty selective process. Yeah. So it's kind of like getting into, I don't know, Harvard or something like that. It's pretty hard. So 33 is true. So 34, a regulatory T cell, a T regulatory cell, is a type of CD4 cell. That's true, right? Then it expresses CD25, which is a transcriptional repressor used by Tregs and is unique to these cells, or it expresses FOXP3, which is blah, 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 same thing as the previous one. So which one's the one that's unique for Tregs? FOXP3, yeah. So correct answers are going to be A and C, so E, right? And this is one of those things that's just memorization, unfortunately. So just try to remember it. I know it's a lot of information, but so 35, we talked about this. It's asking about the positive selection process, and it says positive selection tests whether the blank and blank regions of the TCR combine to a blank. So I did the drawing around here. There it is. So the correct one is going to be CDR1 and 2 with your MHC. Okay? So if you look at it here, HV is the same thing as a CDR, so I'll write it here on the side. HV means hypervariable, CDR means complementary determining region, and they're the same thing. So the HV1 and 2 will recognize the MHC, as you can see here, while the HV3 recognizes the peptide. Okay? Cool. Any questions so far? No? So 35 is C. And 36, when the T cell is screening for the peptide MHC complex on an APC, what is an APC? Antigen presenting cell, yeah, thank you. So do you guys remember which 3 cells are APCs? You should all be singing this. Yes, thank you. So dendritic, macrophages, and B cells. And remember that only which ones can present to T cells, to activate T cells? Dendritic cells, right? So let's write that down. This is what? 36. So APCs, we have macrophage. Okay, that says T cells right there, if you can't read it. But yeah, so your APC is the antigen. It's the presenting cell, antigen presenting cell, and this is asking about when the T cell is screening the peptide MHC. So it says that the T cells LFA1 binds to ICAM1 or 2 on the APC. ICAM3 on the dendritic cell binds to DC sign on the activated T cell, and then SP1 receptor will be suppressed if the T cell is able to successfully bind to the MHC peptide complex. And then A and C are none of the above. So remember, DC sign is a adhesion molecule that's very, very specific for dendritic cells when you're talking about dendritic cells and T cells. So B is going to be wrong because it's saying that the DC sign is on the T cells. And then the SP1 receptor is, so the correct ones are A and C. A because LFA1 is specific for T cells in this case, and it's just like a tight binding protein for adhesion. And it will bind to ICAM1 or 2 on the APC. And then the SP1 receptor, it's that one receptor that tells your T cell to go away if your T cell is not really recognizing anything, or if another T cell recognized what was on that dendritic cell, then you don't want any other T cell to be around the dendritic cell because it's not going to be presenting anything anymore. So the T cell will not be producing that receptor anymore, so that it avoids going away, basically. Make sense? SP1 binds to, I forgot. Just make yourself a little note and look up on the PowerPoints what SP1 binds to, because I am blanking right now. It's one of the chemokines that's on the lymph node. Please, thank you. So then 36 we said is D. Yeah. Yes, it's S1P. I'm sorry. That's a typo right there. I can't hear, sorry. What was it? Mm-hmm.
And macrophages can post up. What do macrophages present to? Because
Oh, okay.
B cells can be involved in that cognate date that they go on.
Yeah, so she's basically asking a good question. Thank you. So she's asking about if dendritic cells are the only one that can activate T cells, then why do you want B cells and macrophages to interact with T cells? And that's because once your T cell is activated, for example, a follicular T follicular helper will be able to go to the follicle and interact with B cells so that they can guide the whole somatic recombination and everything. And then T cells can interact with macrophages to activate the macrophages, for example, and so forth. So Th1s enhance macrophage activity, for example, right? And what Th1s do is that they'll go to a macrophage in the site of infection. The macrophage will have MHC2 loaded with a peptide on it, and if the T cell recognizes that peptide, it's going to activate the macrophage so that it can start killing more of the things that it previously picked up. Does it answer the question? Yeah? Cool. Awesome. So 36 is A and C, D. So which of the following statements regarding the costimulatory B7 protein are true? And so A is saying that the B7 ligand, CTLA4, has 20-fold binding strength less than CD28, which is wrong because it's more. It's a stronger binding that CTLA4 has compared to CD28. B7 on the surface of the T cell binds to CD28 on the surface of the professional antigen presenting cell, and that's wrong, so you don't even have to continue reading the whole sentence. And then C says that when CTLA4 binds to B7, T cell proliferation slows down, which is true. So then the correct answer is going to be C. Does it make sense? B7 is the protein presented that's on the surface of a dendritic cell right next to the MHC. So whenever the T cell is getting activated by the MHC, the T cell will interact with that B7 because the T cell is going to have CD28. So when CD28 and B7 interact with each other, proliferation happens. And when you have enough new T cells, you want to stop proliferation, and that's why your T cell uses CTLA4 to stop proliferation. Okay? Does it make sense? Yes? Cool. B7 is always going to be on your dendritic cell. Yes. So, and it's going to be on your dendritic cell whenever there is no, when whatever the dendritic cell picked up is a pathogen. So if the dendritic cell for some reason picks up a dead epithelial cell and it wants to take it over to the T cells just in case, it's not going to have B7 on the surface. So if a T cell were to recognize that epithelial cell, and because of the lack of B7, your T cell won't get activated. It's actually gonna get, into the anergic state because you don't want that T cell to activate, because if not, you're gonna get an autoimmune response, right? Does it make sense? Yeah? Cool. So 38, right? Yeah. It says a T cell that recognizes self-antigen will most likely be rendered anergic because of the presence of B7? No, it's because of the lack of B7. So let me see the wording on this. Will not be rendered anergic because of the presence. Yeah, so this, I did the wording kind of wrong, weird, just to trick you to make sure you're paying attention. So this is actually none of the above, right? Just because your T cell is rendered anergic because of the lack of B7 on your dendritic cell, or PAPC. Does it make sense? I'm sorry? Because a T cell that recognizes, she's asking why wouldn't it be C that says will not be rendered anergic because of the presence of B7 on the PAPC. And it's because whenever a T cell recognizes a self-antigen, you need to stop that T cell from activating, right? The way you stop that T cell from activating is by not having any B7 on your dendritic cell. So because of the lack of B7, right, you will have that deactivation of the T cell. So it turns anergic, and after some time it just dies off and it won't be able to do anything anymore. Does it make sense? I'm sorry? So yeah, the wording is kind of weird, but if it said will be rendered anergic because of the presence of B7, then C would be correct. Does it make sense now? I'm sorry? Yes, my bad. So it will be rendered anergic because of the lack of B7. If the wording was like that, then it would be true. But please don't let me confuse you anymore. Just think, if there is B7 on the dendritic cell, your T cell gets activated. If there's no B7, your T cell is rendered anergic, right? So 38. So B7 plus MHC T cell activation. If it's only MHC and B7 is not there, right? No B7, then T cell does it make sense now? I'm sorry if I confused you. I didn't mean to. Does it make sense now? Yeah? Okay, cool. So, question? Yeah. Yeah, after a few days it just dies. Anergic means that it's just there, but it's not able to work as a T cell anymore. What do you mean when it keeps recirculating? I'm sorry? Oh yeah, well, I mean, B cells and T cells will recirculate with the S1P, like Lisa was saying. Yeah, but it doesn't have anything to do with the anergic scenario. Yeah, don't worry about it. All right, cool. Just so you know, we have to stop at 6:15 so that we can have space for, you can all leave so that the next people come for the next session. But I probably won't finish it. But like I said, I'll post these as soon as we're done, the answers, okay? All leukocytes or lymphocytes? Are you sure? Okay. So 39, we said 38 was E. B. IL-4 will induce proliferation to what? Th2 cells, right? So, Oh, yeah, sorry. There you go. I'll just start up here. There you go. And that's just memorization, unfortunately. I don't know if he would ask something so specific, but again, anything on the PowerPoints is fair game. This is on the PowerPoint, so if you know it, good for you. Then the disease lepromatous leprosy is a low infectivity disease because it causes differentiation and bias for Th1 cells. Is a high infectivity disease that causes differentiation and bias for Th2 cells. Will have higher concentrations of IL-4, 5, and 10, A and C, B and C. So it is a high infectivity disease, and it will have IL-4, 5, and 10. And it will cause bias for Th2 cells, which will stimulate B cell proliferation instead of macrophages, and that's why it's really bad if you have it. Yeah? No, it's different ones. I can't remember them now. If you go back on the PowerPoint, it's going to have like a little picture that says which cytokines are present. Yeah, but it's going to have bias towards Th1 cells. Yeah. So 41 is E. 42 is asking about the signals that the macrophage needs to activate T cells. You're going to have the primary signal, which is the CD40, CD40 ligand, CD40 receptor, and the secondary signal of the interferon gamma, or the interferon gamma as primary signal and the secondary as CD40, CD40 ligand, or only INF gamma, or only CD40, CD40 ligand. The correct one is B. It's INF gamma and CD40, CD40 ligand. Okay? And again, these kind of things are just straight memorization. Yeah. When an activated naive B cell starts interacting with a T follicular helper, it says that the T cell will release IL-5 and 6 in the medullary cord. IgM is the most abundant antibody that is being secreted by the B lymphoblast. B cell will form conjugate pair with a T follicular helper, which will induce expression of CD40, CD40 ligand, and in turn will activate the NF-kappa B on the B cell, and all of the above. It's all of the above. I did this question so that you know what happens in that scenario. But remember, IL-5 and 6 will stimulate your B cells to start proliferating. IgM is going to be the one that will be most abundant just because it's going to be a naive B cell. That means that it was never activated. And then the conjugate pair will be formed, and you're going to have CD40, CD40 ligand interaction, and CD40, CD40 ligand interaction will stimulate NF-kappa B, and NF-kappa B will make your B cell produce ICAM, which is the protein for tight binding, so that the B cell and T cell can actually stay together for a few days. Yes, question back there? Yes. B and T cell interaction will always happen in your secondary lymphoid tissue. Unless you have an infection in your bone marrow, then you might have interaction of B and T cells down there, but other than that, it's always going to be in your secondary lymphoid tissue, because that's where the activation happens. All right? So 43E. Which of the following statements regarding recently somatically hypermutated and isotype switched B cells are true? We have that we'll have to compete to interact with a T follicular helper. Will die by apoptosis if the B cell is induced to produce the BCL-XL signal, and can turn to plasma cell or memory cell without signals from the T cell. A or C, and none of the above. The one that's actually true, it's only A. That's because B is false, because BCL-XL will actually stimulate the B cell to die, and lack of BCL-XL will induce your B cell to die by apoptosis. And then plasma cells or memory cell differentiation, you always need a T cell for that. You need a T cell to secrete IL-4 or IL-10. All right. So then 44A. Cool. 45. When an infection just began, when we have increasing amount of pathogens in our body, T follicular helper are expected to secrete more blank to induce B cell to differentiate into blank. IL-10 and plasma cells, right? So it's going to be C. Does it make sense why you want plasma cells to be more prevalent during the beginning of an infection? Yeah, because plasma cells secrete antibodies, and antibodies fight the infection. You don't need memory cells at the beginning, right? 46, it says, which of the following proteins are used to slow down the activation of complement? Factor P is used to increase complement activation, and then DAF and MCP are both used for slowdown, right? So that's D. And remember that DAF and MCP, those are used in the surface of our own cells to slow down complement, while factor H and I are also there to slow down complement, but those are used to slow down complement on surface of pathogens. All right? If you don't remember or if you don't know what I'm talking about, just go back to chapter 2 and 3, and it's all going to be there in the nice drawings. It's, very straightforward. Passive and active immunization, you remember what it is, right? Active immunization is when you're making your own antibodies. Passive is when you're receiving antibodies from someone. So in this case, the passive immunization will be Steve Irwin getting antivenom after being bit by a black cobra because he felt like petting the thing, right? I forgot to put the rest in peace for the guy. I feel bad. Anyways, 47. Does it make sense? Cool. Okay. Do we all understand passive and active immunization? Yeah? Cool. Well, it's long. So I'll just tell you the answer for this one, which is C. So naive lymphocytes can circulate through the malt, lymph nodes, spleen, or any secondary lymphoid tissue because they follow a CCL19 and 21 gradient when entering the tissue through the HEV. But once the lymphocyte has been activated in a malt lymphoid tissue, it will follow a CCL25 gradient and will express the adhesion molecule A1, alpha 1 beta 7. That's just because when a B or T cell is activated in your malt, you want it to be dedicated to that place, to that system of lymph nodes. So you just want them to recognize a special cytokine, which is the CCL25, and they'll just express a special type of adhesion molecule, which is the alpha 1 beta 7. You had a question, and then I'll go to you. Oh, it's alpha 4. Sorry about that. So it's alpha 4 beta 7. I'll make sure I change it later, and I'll re-upload it if anything. So then 49. This is from the last test. So when a helminth infection, when you have a helminth infection, the type of response that's more effective, it's going to be what? TH1 or TH2? TH2, right? And that's just because of the type of activation that they do. And then the receptor for IgG, it's going to be Bramble, FCRB, and FCRN. So it's all of them, right? Cool, I actually made it. D, yes. So good luck. Yeah, what's up? You thought it was what? I'm sorry? That's fine. I can. Yeah, thank you.
On 32,
Mm-hmm.
you say it's, I think you said it was D, where it's a fully formed TCR and CD3 and zeta chains.
Let me see. Oh, please make sure you bring back the surveys over here.
I need a survey, and then I have like 2 quick questions.
Hold on, let me answer him really quick and then I'll come back.